Good afternoon. I'd like to welcome our viewers to a conference, a mini conference that is a real prelude to the symposium that's being held here at the Mass General in the next 2 days. The conference is sponsored by the Society for Translational Oncology. I'm Bruce Chabner. I'm the editor-in-chief of the Oncologist, the journal, and also the director of clinical research here at the Massachusetts General Hospital. And I have three of my distinguished colleagues with me to discuss the topic of our meeting and prospects for the future for cancer drug development. The meeting concerns an assessment of where we have come in terms of targeted therapies, personalized therapies for cancer patients, and what are the prospects and challenges going forward. And I'm very pleased to introduce my colleagues, and I'll let them tell you each one about what they do. Jose?
Good afternoon. My name is Jose Baselga. I am the chief of the Division of Hematology and Oncology at the Mass General Hospital Cancer Center in Boston. And my field of expertise is breast cancer and early drug development.
Hi, I'm Dave Ryan. I wear two hats here at the MGH Cancer Center. First, I'm a medical oncologist in the GI Cancer Center, and I direct the GI Cancer Center. And second, I'm the clinical director and essentially working with the operations team to make sure that we meet the goals and the goals of the academic mission of the hospital and of Dr. Baselga.
I should add that David does a great job of organizing the clinical service so that it can interact with the research components of the hospital, and Jose is also very much involved in that, but together they make it possible that we are able to carry out these experimental trials. Finally, the chief of our experimental therapeutics effort is Keith Flaherty. And Keith, please.
Thank you. Yeah, I'm a medical oncologist with a specialization in melanoma, but as Bruce suggested, I have a pretty heavy involvement in early drug development, so came to Mass General just about a year ago to be the director of developmental therapeutics.
So as you can see, the theme of this hospital's research effort is developmental therapeutics, and all of these folks contribute to it in a major way. I'd like to begin the discussion by sort of setting the stage for the history of targeted therapies in cancer, and Jose has been a very important part of that. So maybe you could give us a brief background as to how far we've come in the last 20 years.
So I think, Bruce, a good discussion point is to start defining when did this new revolution with other therapies began? When do we declare that this new era of medicine started? And I think we would agree, all of us, that this happened with the cloning of oncogenes in the mid to late 80s, and with the targeting initially of receptor tyrosine kinases. And you know,
yes, you were very importantly involved in that with John Mendelsohn's laboratory.
Absolutely. So this was a very good time. Basically, we were beginning to develop the concept of targeting these receptors with monoclonal antibodies. This concept was very fruitful in the lab. We were very concerned whether this was going to be something doable in patients and safe. And in the early 90s, we began to see that this was doable. So we began with John study monoclonal antibodies against the EGF receptor. This was cetuximab. Cetuximab was developed then. Mm-hmm. It was called C225 at that time. And we began to see some responses in patients with head and neck and cancer, and patients with colon, and patients with lung cancer. And then there was a cousin to cetuximab, and this was an antibody against the HER2 receptor that was expressed in a proportion of patients. Now we know these are the HER2 positive patients. And this antibody was 4D5 that then evolved into trastuzumab.
There was really a significant difference between the development of the two drugs, because as I understand it, Erbitux began as a therapy for all tumors that express the EGFR receptor. And with HER2, it was clear almost from the beginning that it was a subset of breast cancer patients. So then that introduced the idea that you really had to do the molecular pathology on the tumor to know where to go.
Exactly right. So the model with HER2 was, if you wish, a much more cleaner one. The science was much stronger. HER2 was an oncogene, and it only played a role when overexpressed. So initially, I still remember the phase 1 study, we had any level of HER2 overexpression, what we called at that time 1+, 2+, 3+. but it was clear that only those tumors that had what we know now to be amplification, 3 plus HER2, were the ones that benefit. So that, as you said, that was to me perhaps the first evidence that we need to identify the right patient population. And we had even done simulations. Had we not developed trastuzumab in the right patient population, all the studies would have been negative. And I'm wondering how many of these therapies we have not been able to demonstrate that they are positive or efficacious just because we have not chosen the right
selected
patient.
the right patients.
Yep.
So it certainly has moved forward, even with Erbitux. And David, it's affected your field in particular, the idea that we can now identify the responsive subpopulation. And the first movement in colon cancer really has been the identification of the importance of RAS mutations for Erbitux response.
That's right, and what's interesting about how the field has moved forward over the last 10 years is we went from looking at the receptor status on the surface of the cells to which pathways are actually really revved up. And if you have a KRAS mutation in the colon cancer cell, hitting it upstream of that really doesn't have any effect at all, and in fact probably is harmful to a certain extent, either just because of the toxicity, but the studies that are being done and particularly in the adjuvant setting suggest that it's not the same effect and may actually be harmful when you add cetuximab to a KRAS mutant cell. So there's things going on that we don't understand in colon cancer that when you inhibit with an antibody in the KRAS mutant versus KRAS wild type, it's still a big unknown as to why the KRAS wild types are more responsive for the most part. And in addition, it's very different than what's happened in lung cancer, where you have Tarceva and a point mutation where that gets
activated mutation. Yeah.
Exactly right. So colon cancer in many ways is still way behind our colleagues in lung cancer and in melanoma and in breast cancer. And I think part of the reason is that we're stuck in the antibody world where our colleagues have moved forward with the tyrosine kinase inhibitors.
Yeah, but that too, because you're becoming interested in drugs such as the BRAF inhibitors, which we'll get to very shortly with Keith.
Well, what's dramatically interesting too is someone who's been around now for 15 years is that we all thought 10 years ago that it was going to be the microarrays that identify the different cancer types. We all had a feeling, those of us who take care of a lot of patients, that not all colon cancer is the same. There's probably 5, 10, maybe even 15 different illnesses that are lumped together as colon cancer, and it's true with all the cancers. And we knew that clinically based on how these different diseases developed. And there were people who put down tons of money on the bet that the array or the GWAS was going to be the way we identify these tumors. And what's interesting is it's now being driven by the therapeutic end. If you have an inhibitor of a particular pathway, you're going after that scheme of identifying patients. And just in looking at colon cancer, we now know that based on KRAS, PI3 kinase, BRAF, even now NRAS, and there's probably 5 or 6 more that are important and coming down the pike, that it is a garbage bag term to say colon cancer.
Right.
Nowadays.
And you have all the other GI cancers, each with their own profile of molecular changes. Like I recently saw data on cholangiocarcinoma, which you could probably speak to more than I can.
Well, so things like cholangiocarcinoma have their own set of mutations, even the gastric cancers, the esophageal cancers, the poorly differentiated versus the well-differentiated ones. We're now understanding that. Pancreatic cancer, we used to think that they were all KRAS mutant. There's data to come out recently to suggest that maybe as many as a third aren't KRAS mutant.
Right.
So there's clearly doors that need to be kicked open in each of these other GI cancers that
to explore
these
specific therapies for
exactly.
mutations. So, Keith, this brings us to you. The ultimate test was in melanoma. Take the world's worst cancer and find a treatment for it. And we've floundered for years. And I remember phase 1 studies where we had one response in the first melanoma patients, and it set off a firestorm of optimism, which was very quickly squashed by any future clinical trials. And you came on the field taking on this dreaded disease, and you found an incredible entree into therapy with BRAF.
Yeah, I think Dave introduced the concept really, which is divide and conquer. I mean, I really started with Herceptin and breast cancer. I would say, unlike the development of imatinib and CML, where 95, maybe 99% of that disease is defined by the BCR-ABL translocation and activating mutations in c-KIT, which are so common in gastrointestinal stromal tumor that one could develop imatinib in just based on histologic diagnosis and still capture
so you're
the
really
essence
not
of the signal.
personalizing therapy.
No, but having said that, there was still some refinement of those initial observations. But again, with Herceptin, there was a subpopulation that was responsive, and if you developed it more broadly, it would have failed. I think in melanoma, we'd learned already, from some of what was happening in the targeted therapy era by the time that oncogenes were discovered in melanoma that gave us a unique handle. Yeah. As you say, chemotherapy has underserved the melanoma field relative to colon cancer, breast cancer, even lung cancer for that matter.
So Keith, how did it happen so quickly? The actual mutation, the BRAF mutation, was not discovered until maybe 3-4 years ago.
Well, no, 2002 was the initial discovery, and it took about 4 years from that initial discovery for the first selective inhibitors to emerge for clinical testing.
So Keith, how did it happen so quickly? The actual mutation, the BRAF mutation, was not discovered until maybe 3-4 years ago.
Well, no, 2002 was the initial discovery, and it took about 4 years from that initial discovery for the first selective inhibitors to emerge for clinical testing.
The thing that's different about this inhibitor is it's specific for that mutation. Was it designed for that mutation?
Yeah, it was. Although I would say that it's if you look back at the history of targeted therapy successes, it's actually got a profile of selectivity that's not so different than imatinib. Both as it targets ABL kinase as well as c-KIT. I think many people think of that drug as because it hits 2 oncogenes, it must be very broad. No, in fact, it's not. This is a drug that's a fairly selective RAF inhibitor, but not entirely selective. For example, there's been some notion that if you develop an inhibitor of, in this case, mutated BRAF, you'll then have exquisite selectivity. Well, we see toxicity with these drugs, which we know is due to effects on something other than mutated BRAF.
Does the drug, though, have preferential activity against the mutated candidates?
Exquisitely so.
So that's really a unique feature. Now, the other drugs we're talking about, such as Erbitux or some of the other targeted therapies, were developed against the wild-type oncogene.
Yeah, that's right. And having said that, I think we've stumbled upon a somewhat of a unique observation just really within the past year. After the initial clinical results were starting to mature, it became clear that actually biochemically these drugs have a unique behavior, if you will. So what happens in normal tissues is you actually get upregulation of the so-called MAP kinase pathway in which BRAF sits in normal tissues. Whereas with other targeted therapies, you'll see downregulation of signaling in the tumor and similarly in the normal tissue, and you're looking for a therapeutic index between the two. But actually we see upregulation of the pathway in normal tissue, downregulation only in cells that harbor a BRAF mutation, and we think that actually has a lot to do with the therapeutic index of BRAF targeted therapies in the setting of mutation.
So the inhibition really kills that cell.
It allows you really just to be able to drive the pathway down to lower and lower levels than really have been achievable with other approaches, including even in melanoma, BRAF mutant cancers generally. MEK inhibitors, which are thought to serve a very similar function, don't have that phenomenon of different effects depending on the context. They'll downregulate the pathway in both settings, and the thought is that the dose-limiting toxicities of MEK inhibitors in normal tissue are prohibiting to achieving similar degrees of pathway inhibition.
So a number of issues have been raised by the recent successes in ALK inhibitors in lung cancer, just published last week, BRAF in melanoma, and Herceptin even. And that is, when you develop a very effective targeted drug, and the world knows about it after phase 1, these trials have had 80, 90 patients in it. How do you get it to people quickly?
Yeah, well, I think that the notion is that if we are able to co-develop a method for identifying the patients, so refer to that as a diagnostic as well as the drug, then you set the stage for a very rapid development of drugs, particularly in areas of unmet need. So melanoma is an extreme example. But I would argue that certainly lung cancer is a cancer population that is not terribly well served by available therapies. And so there again, I think if you really refine the population for which the drug is going to be relevant, at least largely enriching there, then even uncontrolled trials can do it. And in fact, that was the case for the early targeted therapy successes.
U.S., we're used to thinking about, or the FDA is accustomed to thinking about overall survival benefit, or maybe time to progression or progression-free survival as endpoints, but even those are considered soft endpoints. Right. In Europe, I think these are a little more flexible. Jose, how are the Europeans thinking about this?
Well, I think one of the problems that we are facing internationally is that there is not a set pattern of behavior, so to speak. So the rules are not clear. So if you're asking me, are the rules of the EMEA, the European agency, consistent? They are not. And I think the process is a process that is evolving. I mean, also to be mindful of the work of the regulatory agencies. Who would have thought the results with the BRAF inhibitors a priori? I think
who would have thought that a phase 1 trial would change the whole thinking about a solid tumor therapy?
Absolutely. So I think what we are all having to deal with is a situation in which we are seeing, in some situations, an amount of benefit that is far greater than even the more optimistic have thought. I'm pretty convinced that the regulatory authorities, the same as we do, when they see this data, we all will adapt. I think that the need, for example, to have a placebo control arm in a subset of genomically defined tumors for which you have a therapy that has an 80% response rate and that results in a long progression-free survival doesn't make any sense. Now, does this make sense when you have a therapy that is potentially incremental? Absolutely. That's where you need a control arm. So I think we are walking into a new era, and I think the name of the game by everybody, by us as clinicians and investigators, and also by the regulatory and by the society, which is the ultimate beneficiary of all this, is going to be a situation of much more flexibility. We are rewriting the rules. Another situation that occurred just 2 weeks ago is the story in prostate cancer, the abiraterone trial. It had a placebo control arm. That's a phase 3 study, placebo control, and then it had abiraterone. And the study was so positive that basically the discussion, this was at the ESMO meeting, said, colleagues, the time to have a placebo control arm in prostate cancer is over. And this has happened 2 weeks ago. So it looks like we're catching up on a daily basis.
We're learning from repeating our own mistakes, maybe, that we have to be very flexible now, and particularly with this idea of profiling patients, selecting the right patient and the right drug, and matching them in a very early point in drug development. So David, your institution and my institution has begun this effort to profile patients. And it's a big undertaking in molecular profiling. Would you tell us something of the scope of what we've done here and what we can expect to happen in the future? And how do the patients react to this?
Well, this was born of the fact that we are a cancer center within a general hospital. I think that's one point that is critical in trying to understand how this all came about. And when I say that, we are constantly in contact with doctors in other disciplines and researchers in other disciplines within medicine. And when you're in a general hospital, one of the...
And by medicine, I imagine you mean surgery and radiation.
Medicine, I mean the medical field in general.
Medical field,
as opposed to being in a freestanding cancer center, it allows you the flexibility to move to where the
patient population is.
Exactly.
So the early patients and the... Right.
And it also takes advantage of the fact that we as oncologists aren't always the smartest guys in the room. And if your pathologists are smartest or your surgeons are smarter, you go to wherever that smart person is. And in fact, at the General, what happened was that a group of colleagues got together, principally John Iafredi in pathology, David Lewis in pathology, Lee Fellison, Daniel Haber, and said, we need to figure out how to do this.
Yes, and the interesting point is that none of them are major practicing therapeutic interventionalists. They are people pathology, basic science, and laboratory medicine. So they perceive this as being game-changing.
Hugely game-changing. And I think that they were taking their cue from the clinicians who were saying, we need to do this. You need to get us these tests. How are we going to do this? And they had the vision to say, well, why are we just looking at these mutations? Let's look at as many mutations as we possibly can at that particular time. And so it first started in lung cancer, where we started to take everybody with metastatic disease where there was really an unmet need, which was we needed to know what the EGFR mutation status was of those particular patients. And we started doing all of the metastatic lung cancer patients. We didn't have the capacity to do everybody with metastatic disease. And so when an academic mission comes down from on high, we take it back to the operations folks, and we have to shift resources to make that happen. Gradually, as time went on, we were able to open it up to the GI world, to the melanoma world, to the breast world, and on down the road, so that our ultimate goal is to have it available for everybody with metastatic cancer. But what was interesting, and I think speaks to how important this is what grew out of that was the ALK story. So the ALK story wouldn't have happened had we not had the vision of the leadership and not had the flexibility to do what we did. And in fact, Eunice Kwok, when she was, the great story with the ALK story is that Eunice Kwok and Jeff Clark were running a phase 1 study of crizotinib, and
which is a MET inhibitor.
Which is a MET inhibitor. And you say Eunice is my colleague in the GI Cancer Center, and Jeff, of course, is a long-standing colleague in the GI Cancer Center, has been around the phase 1 world for 25
Almost
years.
Almost as long as me.
Yeah, yeah. And is well known within the phase 1 world. And so Jeff and Daniel Haber were at a meeting, and all of a sudden it came out that not only does it hit MET, it actually hits ALK. And the folks who were presenting had no idea of the importance of that. So they come running back, and they say to John Iafredi and Leif Ellison, can we add ALK to our big snapshot panel? Lo and behold, they add ALK, and they shifted all of the resources from looking at this as a MET inhibitor in stomach cancers to looking at all the
ALK inhibitor in lung cancer.
I mean,
well, there had been an important paper published, I guess
correct.
It was in 2007, and then some subsequent papers showing that a very small subset of lung cancer patients, 3-4%, had ALK fusion mutations that activated the gene, and that they even showed in laboratory animals, if you used an ALK inhibitor, that you could cure animals, you could make the tumor disappear. So it was a very logical thing to say, well, let's look for this subset of patients as part of the phase 1 trial.
And what's fascinating to me on an operations standpoint is we actually had to divert resources to this effort and slow down the effort globally within the cancer center, because we knew we were proving a point here, that if we proved that being flexible and going after these mutations as they come up and adding them to the panel was the right way to go, that in the end it would benefit all of the folks with every different type of cancer. And so it slowed down our ability to offer this genotyping, this global genotyping, to the rest of the cancer center, but we knew that if we were able to complete this study and prove it, that it would be such a home run. It would have a far-reaching impact, and in fact it did.
Don't you think the more powerful element of Snapshot, and this genetic approach, which there are other platforms that could accomplish it, and to me the reason why cancer centers everywhere need to adopt something like it is not just this discovery and quick sort of clinical research translation, but it's the fact that increasingly we find that mutations are not private within a certain histologic subtype of cancer.
Right.
And so without translocation there is actually some spread across cancers, and a few other instances, but I think some oncologists are used to the idea that BCR-ABLs in the leukemias,
that's right,
and KIT mutations almost exclusively in gastrointestinal stromal tumor, but BRAF and RAS and PI3 kinase, a story that Jose can expand upon enormously, a very hot area. These spread across cancers, and the issue that we ran into with BRAF was, yes, it's most common in melanoma, so melanoma as a clinical arena will lead that area, but you find them in 2 to 3% of lung cancers, and it's just, as the BRAF therapies mature and become standard treatments, it's just not efficient to screen for something that's so infrequent within a given population. And a panel like this allows you to, if you will, passively detect them.
Yeah.
Exactly.
You really raise an interesting question, which I'll pose to Jose. That is that one of the really promising areas that's going to be discussed in the next 2 days here is the attempt to address mutations in the PI3 kinase pathway. And we've even got drugs approved. We got the mTOR inhibitors. But the research to this point has not been focused on understanding the specific mutation in the patient and then putting the right drug in that patient. Jose, are we going to see movement in the
Absolutely.
PI3
So the
kinase?
PI3 kinase is one of the genes that is most frequently mutated in cancer in a variety of tumors. So it's present in breast, it's present in glioblastoma, GI tumors, others, lung cancer. So clearly, and the pathway is extremely important. And you have mutations along the pathway. So you have mutations.
Multiple points.
Yeah, absolutely. So it's PI3 kinase, it's AKT, it's TSC1, TSC2,
IGF receptor or not?
IGF receptor as well, P10. So the pathway is importantly, and there are mutations at every single step in the pathway, which to us is an indication that the pathway is important. That's a rule of thumb, you know.
Yeah.
When you have activating mutations in cancer, those are usually important. So yes, we have shown that you can target the pathway with the mTOR inhibitors. We did the phase 1s and we began to see some responses. But it is clear that the rapamycin analogs, the mTOR1 inhibitors, are not as optimal as the PI3 kinase inhibitors themselves. Now, the biology is complex. We have multiple types of compounds. So we have pure PI3 kinase inhibitors. We have PI3 kinase and mTOR inhibitors at the same time. We have PI3 kinase alpha. There are several subtypes of PI3 kinase, and some of the compounds are pan inhibitors. Some others are isotype specific. So we are working on that very carefully. We are part on one of these large Stand Up to Cancer. We have conducted the initial phase 1 studies that we presented at ASCO this year. And here at Mass General, I don't know how many protocols we have, I stopped counting, targeting PI3 kinase from every single possible angle. I think the future will be, again, to identify the patient population. That's why we are snapshot. We have a snapshot.
The logical patient population.
Yes, so we have to study the ones that are mutant. That's point number 1.
Yeah.
But point number 2, something that we are learning, and this is a topic for discussion in depth that is going to be a main topic for this symposium in the next two days, is the need to use combinations. Because what we are learning is that the PI3 kinase is such an important pathway that if you block it, cancer cells have the means to activate compensatory pathways as a way to survive that. So it's not going to be that easy. You are not going to get away by blocking.
You're not going to see tumors melt the way you see them melt with BRAF.
It's not. The compensatory pathways. So this is like Boston. Basically, if you block the MAS spike, there are back routes.
The storage drive.
Yeah, the storage drives of the world. And so we have to block those too. So I think it's going to be, but it's going to be very exciting. And we have some indications. For example, you alluded to the insulin-like growth factor receptor. We have now data in the phase 1 study, Bruce, if we block mTOR together with IGF-1 receptor, we are seeing a huge response in luminal B breast cancer, which is a very tough subset.
Yes.
So, and now working with Keith, we're going to be doing some combinations with MEK inhibitors and PI3 kinase. Very powerful rationale to do that.
Yeah.
So I think
it's going to be more complicated. It's going to take more than one drug.
It's going to,
some of these.
Yes, it's going to be apparently. I think apparently it looks like unbearably complicated, but I think that like always in medicine, it appears to be very complicated, and then it becomes very simple. So I would hope this will become simple.
Well, and I would point out that in some fields that combinations have accomplished a great deal, such as in HIV. Where would we be if we were still in single agent therapy of HIV? So it may be the same situation, that the more complex cancers, the ones that are not solely dependent on one mutation, are going to need combinations of drugs. Again, it's going to have to be based, though, I think, on molecular profiling and understanding the individual patient, which is a challenge for us these days because there's all these concerns about the cost of medical care. But I would make the point that if we select patients right and use our drugs for patients who are actually going to respond, that we're going to save money and we're going to extend lives in a way that we couldn't when we were using the shotgun approach of chemotherapy. But that's our challenge. We have to not only be effective, but we have to be cost effective in doing all of this. Well, I'd like to close the session by asking each of our panelists a question about what is the next important trial that you see being done in your area of interest. Keith, we'll start with you.
I think we've touched upon it before. The notion that we develop exquisitely selective targeted therapies where we identify, if you will, a proof of concept population, a population where the target is really critical for cell survival, but then look for ways to try to partner, if you will, in settings where it's going to take more than one drug. Certainly one can point to instances, but I think the MAP kinase pathway and the PI3 kinase pathway together, this is going to be a question that's going to take some time. It's likely there'll be different sorts of combinations with agents within the PI3 kinase pathway in particular to complex with agents in the MAP kinase pathway. And it's not going to be a one-size-fits-all strategy. It's not now with the first generation of single-agent targeted therapies. So as you suggested before, I think we're going to need to be profiling as we go in and as we come out of these trials to understand in whom did they work and why.
So are you thinking specifically about the RAS mutant melanomas?
Well, that's critical. I mean, RAS has been one of the more bothersome targets, if you will, being so commonly mutated in cancer, approximately 30%. One of the earliest discovered oncogenes, in fact, and yet not a candidate for direct drug targeting based on available technology. And until that problem is solved, we have to deal with the so-called RAS effector pathways. And we've been talking about
the downstream.
Exactly. So that's where the MAP kinase pathway and the PI3 kinase pathway come as the two best studied of them. But there are several others still. And trying to understand in the setting of RAS mutation which pathways are most important in individual tumors. This is going to go to another level in terms of profiling that we've talked about today. Right. But I would just emphasize that I think there was an era, brief as it may have been, in this only about 10, 12, 13-year-old targeted therapy history where drugs that were somewhat promiscuous were considered potentially potentially thought to be good because you might hit the intended target and pick up a few more sort of by happenstance. Yeah. But I think we're done with that era. We actually started with drugs like Gleevec, which are very, very selective in my view. And now we're back to that. If we're going to build combinations, we need very selective agents because the toxicity of combinations otherwise will be unbearable.
Yeah, yeah. David, what's your favorite idea for the next 3 years?
For the next 3 years, I think the scourge of pancreatic cancer and KRAS mutant colon cancer, I'm very excited about the combination pathways, particularly the MEK plus the PI3 kinase pathway inhibitors of really making some headway, at least in a few of these KRAS mutant subtypes. I would say the other thing that we need to start thinking about too is curing patients. And there was a cautionary note in the GI cancer world this last year when Erbitux or cetuximab for those folks who had KRAS wild type tumors actually didn't help at all for those patients with stage 3 colon cancer.
In the adjuvant study.
In the adjuvant study. It was very upsetting. We thought we were going to see something very similar to what the Herceptin story was in breast cancer. And I think we're going to have to be very careful about our old ideas of it's the same disease, it's just stage 3 and stage 4. They may actually be two totally different diseases with very different molecular profiles. And I think we have to move the molecular profiling into the adjuvant setting, just like we've moved it into the metastatic setting. And then the flip side of that is we're still not curing anybody with EGF mutant lung cancer or ALK mutant lung cancer.
As much as we celebrate.
As much as we celebrate it. Right. And if we're killing off 99% of these cells, what's preventing us from killing off the other 1% that hang around for 6, 12, 18 months and then come roaring back? And so we need to figure out how to attack those cells that are being left around that somehow develop this resistance.
So there are some very nice papers at this symposium addressing resistance and molecular studies of mechanisms of resistance in lung cancer patients in particular. Jose, what are your thoughts about breast cancer? What are you putting your money on in terms of the horse race to the finish line?
So, I mean, there are a number of things that are, to me, are very exciting. The combination with IGF receptor and mTOR, very promising in low-metal breast cancer. Combined therapies against HER2 and HER3 in HER2-positive disease, very exciting. The possibility of targeting the EGF receptor in triple-negative breast cancer. That's where the thing started. We initially thought that EGFR was going to be a target in breast cancer, and now we have some data in support of that. So all this is very exciting. But to me, if I may, there is one thing that keeps me even more excited, and that is to be here in this place at the Mass General and to see what we can do here. I mean, I think we could really continue to lead this way of new targeted development, and we can and we will incorporate imaging. You know, imaging is going to be a key player here. New technologies, biomarkers, to show true target engagement. Are we hitting BRAF as hard as we should? Are the differences between the different compounds that are being studied due to different in target inhibition? Can we measure that? We should be able to measure. Bringing new platforms such as the circulating tumor cell platform that gives us a promise of really interrogating the tumor, if you wish, on a daily basis. And then bringing the cell line screening, the Center for Microtherapeutics, that would allow us to screen multiple thousands of cell lines and identify relationships between genotype features and specific drugs that are active. So I think this is what really makes me excited. We are building these platforms in which we could have, hopefully, an ALK story every few months if we are working in the right direction. So, and it's going to be a very exciting next few years. I'm really looking forward to this.
That's a wonderful way to close this. And let me say that I have confidence that if we use science well, that we can address the problem of cost benefit, even in this current climate of fiscal restraint, that we will extend lives and make therapy more cost effective by using science. Thank you very much.